Welcome everyone. Today we are going to learn about the role of non-gated ion channels in the generation of resting membrane potential. At first, understand these four important points. The first is ATP driven pump. Second is ion concentration gradient. Third is non-gated ion channels. And the fourth and the last is electric or membrane potential. We can also call it voltage gradient. So we have to understand at first these 4 points. Then we can understand what is resting membrane potential and the role of non-gated ion channels. From the first, I want to mention that ATP driven pump, I am giving one example, that is sodium and potassium pump. Now this pump actually hydrolyzes one ATP molecules. It hydrolyzes one ATP molecules into ADP and PI. So it requires hydrolysis of ATP to get energy, and it actually export. Suppose this is the cytosol and this is the exterior of the cell. It export 3 sodium out of the cell, and it actually import 2 potassium ions. 2 potassium ions inside. Okay. Now, why I am mentioning the role of ATP driven pump? Because this pump actually helps in the generation of ion concentration gradient. As you can see, this pump is actually exporting sodium out of the cells, so the concentration of sodium outside will start to increase, and simultaneously the concentration of sodium inside in the cytosol start to decrease. And the opposite occurs as because this pump actually importing potassium ions, so the concentration of potassium ions inside the cell increasing, and the potassium ions concentration in the outside is actually decreasing. Now the concentration difference of ions in the exterior and the cytosol is called ion concentration gradient. So there is a difference in the ion concentrations in the exterior and the cytosol, and we call it ion concentration gradient. I'm giving just one example, sodium potassium pump. There are so many ATP different pumps present that actually generate this ion concentration gradient. I'm giving one example here. In case of mammalian skeletal cells, the exterior of the sodium concentration is 144 millimolar and inside is 12 millimolar. By the working of sodium potassium pump, as you can see, the outside sodium concentration is high than the inside, and in the skeletal muscles also the potassium concentration inside is actually 155 millimolar, but outside is actually just 5 millimolar. See, there is a difference of the ions concentration, and that's we call ion concentration gradient, that is generated by ATP driven pump. Now this is very important to understand this resting membrane potential. This difference is very important. We also call this ion concentration gradient as chemical force. Remember the name, chemical force. Okay, chemical force. And this electric or membrane potential, that means the voltage gradient, we call it electric force. Electric force. Remember these two terms, because these two forces actually helps in the transportation of ions. Okay. So see, at first the background is set by this ATP driven pump. Here I am giving examples of sodium potassium pump. So in the plasma membrane of all cells. Now I am not only talking about the resting membrane potential of excitable cells like neurons, muscles, not only these excitable cells. I am talking about all animal cells. In case of all animal cells, there is resting membrane potentials. Now for the generation of this resting membrane potential or electric membrane potential, at first I will talk about what is electric membrane potential. There is a role of non-gated ion channels. It comes third point. See, here in the plasma membrane there is potassium channels present, sodium channels present, chlorine channels present. Okay. Basically, scientists have discovered that these three ions are actually play important role in the generation of resting membrane potentials apart from calcium, magnesium or other ions. These three ions are very important for the generation of resting membrane potential. Now, among them, scientists also observed that basically the potassium channels actually play important. So many potassium channels actually plays in this generation of resting membrane potential, and very few sodium channel and chloride channels actually take part for the generation of membrane potential or resting membrane potential. Now these channels, I'm talking about these potassium channels, is called leaky potassium channels. Leaky potassium channels or resting potassium channels. Remember this. Why it is called leaky potassium channel or resting potassium channels? Because in the resting conditions, this channel is actually open in conformations. A channel can go into two different states, open and close. But this channel is open during this resting membrane potentials. Many potassium channels actually open. Very few sodium channels and chloride channels are open during this resting membrane potential. So these ions actually play a little role, but majority is done by the potassium channels. Now the channels is also called, this potassium channel is also called the non-gated channels. Non-gated. Why it is called non-gated? Because we know that channels can be divided into two categories. One is non-gated and another is gated. There are three categories. One is voltage gated, there is ligand gated, and there is mechanical gated or mechanically. Now what happens for the gated channels? The closing and opening of the channels depends on the outer force, like it could be a voltage change, it could be the binding of ligand or mechanical stress. So the opening and closing depends on external force, but in case of non-gated ion channels, no requirement of external force is important. They are already open, so that's why they call leaky. As because they are open, they called leaky. Don't confuse the terminology. This leaky channels is also called resting membrane potential. That means these potassium channels are resting in condition. No. As because they generate the resting membrane potential, that's why it is called resting potassium channels, but they are not resting. They are open in conformation. Remember this. These channels are open. And through this, what will happen? See, channels actually transport ions from higher to lower concentrations. Remember this. I have mentioned in my previous videos that channels transport ions from higher to lower concentration. Now, see, where is potassium ions is higher? See that in the cytosol, potassium concentration is high and outside is low. So potassium will, what will occur? Potassium will move out of the cells. So potassium ion will move out of the cells. Okay. In case of sodium, what will happen? Sodium, as because outside is high, so very few sodium, as because very few sodium channels are open, so few sodium will come inside the cell. The sodium potassium pump is also actually electrogenic in nature. Remember the term electrogenic. Electrogenic. Now come into this point, the electric or membrane potential. This sodium potassium pump is also electrogenic. Why? Because see, here it actually removes 3 sodium out of the cell and import of 2 potassium. That means it is actually exporting more positive ions outside and importing less positive ions. And that's how in the inside, that means in the cytosol, there will be more negative charge ions will be present, because always there is a balance. If you move more positive ions, what will happen? Negative ions will be left behind. So as because one more positive ions are actually exporting, so one negative ions will be left behind. And the simultaneously, as because more positive ions are moving outside, there will be a positive charge starts to occur outside, positive charge. One positive charge, okay. But one thing we have to understand, this is obviously the membrane potential or voltage gradient. What is voltage gradient? In case of electric or membrane potential or voltage gradient, that means there is a difference in voltage outside and inside, and that is actually starts by this sodium potassium pump. But remember, a very fraction of amount is actually generated by the sodium potassium pump. A very low fraction of voltage gradient is generated by sodium potassium pump. When potassium channels open in conformation in the resting conditions, normal situation, resting means unstimulated conditions, when animal cells in unstimulated, then many potassium channels that are open, through this potassium ions actually move from inside to the outside. That actually generates huge amount of membrane potential. Huge amount of membrane potentials, membrane difference. That means potential difference occur. Huge amount of potential difference occur inside and outside, as because more potassium ions are actually moving out of the cell. That actually generates the electric or membrane potential or voltage gradient. Now, how could we measure this? If you put micro electrodes inside a cell and outside, if you put another microelectrode, and if you connect these two microelectrodes with a voltmeter or potentiometer, you will get a reading. And scientists have observed that the measurement of this resting membrane potential, that means when the cells in at rest condition or unstimulated, it is approximately, the resting membrane condition is approximately minus 70 millivolt. Remember the value, minus 70 millivolt. But obviously they have observed that this resting membrane potential can vary from minus 20 millivolt to minus 200 millivolt also, and that depends on the cell type and the organisms. But generally it is minus 70 millivolt. Remember, this is the resting membrane potential. Okay, now one thing I want to mention, now this is the overall understanding that aqueous ATP driven pump generates the ion concentration gradient, and then this ion concentration gradient is actually utilized by these non-gated ion channels. They are open in form, so as because many potassium is open, so the contribution to this electric or membrane potential is actually goes to this potassium channels. So more potassium channels actually goes out of the cell, and that generates the membrane potential, resting membrane potential. Now one thing I want to mention here, that this ion concentration gradient, we call it chemical force, and this electric membrane potential or electric force, both these two factors when combined is called electrochemical gradient. So when this ion concentration gradient and the membrane potential or electric potential togetherly work, as in this case, in case of sodium and potassium ions, as you can see, there is ion concentration gradient also, there is membrane potential is present also. So when ion concentration gradient and membrane potential combine, we call it electrochemical gradient. So here two forces working together, chemical force and the electric force, driving the transport of ions. This electrochemical force is also called ion motive force. It is also called, similarly we can call it ion motive force. Two scientists, I'm writing here the name of these two scientists, remember their name, Alan Hodgkin and Bernard Koch. In 1949, they performed a simple experiment. Remember this name, Alan Hodgkin, he has actually performed so many groundbreaking research on the nerve impulse propagations. Try to understand what is the reason behind this generation of resting membrane potential that is approximate to 70 millivolt. They are actually getting the value by putting the electrode. Now they ask the questions, why this value, why it is generated? They try to change the external concentrations of ions. Okay, they try to understand which ions is responsible for the generation of this membrane potential or resting membrane potential. So when they change the concentration of sodium and chloride in the exterior, so they actually use squid actions. So they change the outer concentration of sodium, chloride or other ions, and they found a small change occurs in the resting membrane potential. A small change occurs. But when they change the concentrations of potassium in the outside, they find that significantly this value is changing. What they do, they actually increase the potassium concentration artificially outside. Now, if the concentration of potassium increases outside, what will happen? Less amount of potassium will move, because the difference is actually getting low. As much as the difference is getting low amount of potassium will move, and that's why the membrane potential will drop. The resting membrane potential will drop. They found that the value of the resting membrane potential is actually dropped into minus 70 millivolt to, suppose, minus 60 millivolt. It is becoming less negative. Less negative means less value. And when they observe another fact, that is, when they change the potassium concentrations, just like the inside concentration, that means if we change this potassium concentration to 154-55 millimolar, they find that the resting membrane potential becomes 0. That means there is no net movement, because the ion concentration gradient is not present there. There is no movement of potassium, there is no generation of resting membrane potential, and it gets the value of 0. By this experiment, simple experiment of Alan Hodgkin and Bernard Karch, they discovered that potassium channels is important and potassium movement is very important by this non-gated or leaky potassium channels to establish the resting membrane potential.